Hello, everyone. I'm Lydia Lynch, and I just want to thank Philip for organizing this, for inviting me to give a talk at this, but also for putting this together last year when we really needed it and we didn't know if we'd be going to another conference. And so it's really nice to see that he's continuing this this year as well. So my talk is going to be on how systemic metabolism dictates immunity. And so, immunometabolism can mean different things to different people. So on one side, there's cellular immunometabolism, which is the bioenergetics of the immune cell and how the different metabolic pathways that the immune cell uses controls their different effector function. And then on the other side, there's systemic immunometabolism, which is the unexpected role that the immune system plays in regulating systemic metabolism. So this can be, for example, the adipose tissue immune system, how the immune cells in adipose tissue, they can dictate and they can regulate adipocytes and adipocyte storage and fat burning. And then this affects obesity, type 2 diabetes, insulin resistance, but also the effect that these systemic metabolic states have on the immune system and how this is linked to different immune disorders. And so my lab studies both sides of immunometabolism because we really study how different metabolic states in the body affect the immune cells because different systemic metabolic states, for example, obesity or the differences that happen in the tumor environment or in the whole body, for example, during cachexia, actually change the fuel and nutrients that are available to immune cells. And then that dictates different immune pathways that are used, or sorry, different metabolic pathways that are used in the immune cells, which dictates their different function, which then comes back, depending on what cytokines they produce, comes back to play a role in systemic metabolism again. And so this link here is what I'm going to talk about today. And so how do we do this? I'm just going to briefly describe the patient. We study this in humans and in mice. So we collect samples from patients at the weight management clinic, in the diabetes clinic, and those patients undergoing bariatric surgery, both in Ireland and in Boston. And we collect serum from patients for metabolomics. We also collect blood, adipose tissue and liver when we can. And we do lymphocyte functional, immune assays. We also do transcriptomics and metabolomics on the samples. And we can then confirm this in larger data sets from the biobank. And in mice, I'm the director of the metabolic core, and so we can study mice in these metabolic chambers where we can look at really different, we can change the environment to study, say, cachexia or a short-term high-fat diet, long-term high-fat diet, weight loss, exercise by looking at their whole body metabolism, their response to exercise, their food absorption, their fat distribution, and obviously seahorse to measure the cellular metabolism when we make all these metabolic changes. And so again, we collect serum, we collect bone marrow, lymph nodes, spleen, liver, adipose tissue, white and brown, and we look at the functional outcome of immune cells, the whole tissue function, and single cell sequencing, whole body or whole tissue sequencing, and we look at it in these metabolic states, as I just said. And so the goal of all this is to really get a picture of, by looking at, first of all, the whole body metabolism through the metabolic cages, and then at a deeper level at the, what's happening to the metabolome, what's happening to the transcriptome of the cells, at the cell level and at the tissue level, so that hopefully we can put all of this together and see the major pathways that are changed in obesity or in different metabolic states. And so the areas of interest where we're studying this interdependence of systemic and cellular immunometabolism is obesity. So that's the main one that we're studying. What does obesity do to our immune system? And what impact does this have on infection or cancer or autoimmunity? We also study cachexia to see if there's any, we're studying if there's any immunometabolic targets here, although I won't talk about that much today. cachexia is a metabolic switch that happens in, for example, cancer cachexia, or it can also happen in HIV or chronic heart failure. And the whole body switches to fat burning. So we're studying that from an immunometabolic point of view. And also more recently, neuroimmunology, looking at immunometabolic targets here, mostly at the moment around IL-17. And so as an immunologist, I became interested in obesity about, over 12 years ago now, because at the time everybody knew, obviously, obesity is associated with many different diseases, particularly known as the ones in blue: diabetes, cardiovascular disease, insulin resistance, fatty liver disease. But at the time, it was really becoming obvious that it was also associated with a lot of immune-related diseases, such as respiratory infection. We've seen this during the H1N1 pandemic. It was clear that patients with obesity had increased risk of severity and hospitalization. We obviously see it again now with COVID, especially in young population. Patients with obesity that get COVID have increased risk of severity and mortality because of COVID. At the same time, vaccinations are less effective. There's less of a robust immune response to vaccinations in patients with obesity. Also, there's increased risk of psoriasis, multiple sclerosis, rheumatoid arthritis, allergy, asthma. There's many different immune-related diseases associated with obesity, and also cancer, many different cancers. And so cancer is what I'm going to focus on today. So there's about 14 cancers that are increased risk with obesity. And some, for example, endometrial cancer, half of endometrial cancers are due to obesity. Worldwide, in total cancer cases, 500,000 cancer cases per year are due to obesity. And despite this, despite us knowing it, when in a survey of the general public, even when prompted, most people, 84% of people, did not realize the increased risk of cancer associated with obesity. So we really need to do a better job of communicating that. And so where did we start? We started to look at NK cells because they are the first line of defense against infection and cancer, both of which are increased in obesity. And so this is published 2 years ago now, so I won't go into much detail, but it just serves as an introduction to the talk. And so we started to look at NK cells, mainly in humans, in patients with obesity compared to age and sex-matched lean patients. And we found basically that NK cells are metabolically paralyzed in patients with obesity. So this means when they see a tumor target, they cannot turn on their metabolism, their metabolic pathways, they can't upregulate them, and this affects their function. So just to give you some key highlights from how we found this, we sequenced NK cells from lean and obese people and mice. And the first pathway, the top pathway that we found was downregulated in NK cells in obesity, was cytotoxic genes. So there's a loss of granzymes and perforins in NK cells in obesity, which is obviously the killing machinery that they use to kill. And then on the other side, the most upregulated genes were related to lipid metabolism. So there was CD36, which is a scavenger for fatty acids, many fatty acid binding proteins, LDL receptor, lysosomal lipases, and CPT1A and B. And so interestingly, these are all PPAR target genes, and PPARs are the transcription factors that control lipid metabolism. So this suggested that in obesity, in a high lipid environment, NK cells turn on lipid metabolism, probably because they're taking in the lipids. So we wanted to check this, and just isolating NK cells straight from blood of patients with obesity or who are lean, we see if we stain with lipitox for a neutral lipid stain, that there is more lipids inside, or more lipid positivity in NK cells in obesity. And it wasn't that the NK cells were really bigger. So it wasn't because they had more lipid membrane. Here's an NK cell from lean, this is the lipid membrane. really in obese, you could see lipid droplets inside of the NK cells, which are not usually present in NK cells. And so this kind of reminded us, NK cells, or probably all immune cells, are what we eat. So if you put a cell with lots of, mainly different fuel supplies, for example, more amino acids or more fatty acids, they'll adapt their metabolic programs to take on the whatever fuel is available and reprogram their metabolism to potentially use this fuel. And so we wondered, can NK cells use lipids then? Are they able to use lipids for metabolism and function? And so to do this, we took NK cells from lean patients and we gave them different fatty acids in culture. And we tried palmitic acid and oleic acid and a combination of the two. And we find, first of all, when you give NK cells lipids, they take up lipids, so they become more positive in lipid staining. But we found that if we look by seahorse at ECAR for glycolysis and oxphos, we see that this is the normal response. This is in activated NK cells in black. They've upregulated both glycolysis and oxphos. But when you treat them with lipids like palmitate or palmitate and oleate, they're not able to upregulate glycolysis or oxphos. I suppose the glycolysis was not that surprising, but it suggests that they're not really doing fatty acid oxidation either. And so then we looked, that was cells treated with lipids. Then we looked directly from the blood with Andy Hogan in Ireland. And we found that when you take NK cells directly from people who are lean or obese and run them on seahorse, this is NK cells here in the dotted black line, unstimulated versus stimulated from lean. And they've upregulated both glycolysis and oxphos, but in NK cells from patients with obesity, they don't upregulate either glycolysis or oxphos. So they really are just metabolically paralyzed. They're not dead. The NK cells are not dead. They're not dying, but they're unable to function properly or turn on metabolism. And when we looked inside, they had much less ATP as well. And so next we looked inside the cell. And so here is to see if this was related to their loss of function as well. So here, for example, is NK cells from control lean patients. And this is LAMP1 staining for their lysosomes or their lipid granules. Lipid tox, there's not much lipid droplets inside of them, and perforin, which is found in the lipid granules. So the marriage here is perfect. But when you treat NK cells with lipids, you find that, so here we can still see lipid granules, but now you can see lots of lipid droplets inside the NK cells. And there's a loss now of perforin inside of these cells. And so here is an interesting image. There's 3 NK cells here. 2 of them have taken up lipids, but this one has not taken up as many. These 2 that have taken up lipids have no perforin, whereas this one who hasn't taken up so much lipids still has perforin. So there is a loss of cytotoxic machinery when an NK cell takes up lipids, and also a loss of their metabolism or their metabolic, their ability to turn on metabolic pathways. And we think that these 2 are linked, potentially because downstream of the metabolic pathways like MYC and mTOR is the transcription of interferon gamma and granzymes. Obviously this then impacted their function if they don't have any perforin. So if we put them then with tumor targets in vitro, they are not able to kill them. And this was Xavier in my lab that did this work. He's a cell biologist, and so he really loves the lysosome and how the cell moves on the inside. And so he wanted to know, why do you actually need these metabolic pathways to kill? Like, where is the pathway important? And so he did this by imaging. So this is a lot of this information came from Jordan Orange's work on NK cell cytotoxicity. And so here you can see the microtubule organizing center and all the lipid granules in the cell. And when a cell sees the target, for example, the tumor, it forms a synapse by rearranging its actin filaments. This sends a activation signal to the cell, and the mTOC pulls all the lipid granules together, and then they deliver this to the synapse. And this is really very important. This kind of very targeted cytotoxicity, because if they just release their granules all over the place, they won't kill the target. And so you can image this. And so this is what Xavier did. So he used perforin stain to image the lytic granules, pericentrin to image the mTOC, and CD2 to image the synapse. And so first of all, we see when we put NK cells here, it's a small cell with the tumor cell. In vitro, it's able to see the tumor, it attaches on, it forms a strong synapse, which we can see by CD2, and the pericentrin is so the mTOC is at the synapse for release of the granules into the tumor for the tumor cell to die. However, when you pre-treat the NK cells with palmitate, they still are completely viable. They're still able to see the tumor, they're able to migrate to the tumor, they're able to attach, they're able to form a synapse, a strong synapse here, but the pericentrin does not move. So, so the lytic granules do not move to the tumor. And then if we just take NK cells directly from patients who are lean or obese, here is just directly ex vivo. NK cells have seen the tumor, they have formed a synapse. This is the pericentrin, this is the mTOC. But in obese, this is 3 NK cells attached onto this tumor cell, and in all cases, the cells have formed a synapse, but the pericentrin has not polarized to the synapse. And as a result, because NK cells are serial killers, they don't not just kill this tumor, or potentially eventually they will kill it, but it takes much longer. That they're prevented from killing this because they can't mobilize their lytic granules to the site, but also when they do kill the tumor, it gives them a signal to detach and kill again. And so if they don't get that signal, then they don't go on and kill other tumors either. And so we think this is the part of their metabolic pathways is very important for mobilizing their organelles to kill. And so I haven't put it in here, but when we block mTOR or glycolysis, we don't see this polarization either. So we believe that glycolysis is required for this process. And in obesity, the inability to turn on glycolysis results in their inability to kill. So then on from this, Lydia wanted to look more about this in vivo. And so we had to obviously turn to mice to do that. And so she did two different two different cancer models, B16 and MC38. And in both, there's an increased tumor growth in mice that are fed a high-fat diet, so obese mice. This is particularly true in MC38, which is more T-cell dependent. And so what she found, which I won't go into now, is adaptive transfer of NK cells from obese mice or NK cells treated with lipids were unable to control this tumor growth when they were adaptively transferred into mice-bearing tumors, unlike NK cells from normal standard fat diet mice, which could reduce the tumor growth. But from this work, she was originally more interested in T cells, and so we looked at the other components of the tumor infiltrating compartment, not just NK cells, and we found it was really across the board, it was all the cells that were affected by obesity. So in B16 and MC38, CD45 total numbers were decreased in the tumor of obese mice, both models. CD8s were also decreased in both models. CD4 very much decreased in B16, and NK cells were decreased in both models. And so then she looked a little bit more into T cells and found that, the tumor environment is already immunosuppressive, but it was even more immunosuppressive in mice that were obese. So they had the CD8s had less proliferation in the tumor, much less interferon gamma and granzyme B, less amino acid transport, which we did with the kynurin assay, and less phosphorylation of S6, which is in some way a surrogate of mTOR signaling. And so it wasn't just NK cells that are defective, but also other anti-tumor immune cells. And she didn't find that this was because there was more exhaustion in the immune cells, in the T cells. So in our hands, the PD-1 levels from going from lymph node into the tumor, they go really high. So they're already, so high they couldn't go any higher. So there was no increase in PD-1. They were both the same in lean and obese mice. And if anything, there was slightly less PD-L1, so it wasn't that there was more exhaustion, but potentially there was less activation in the first place. And so potentially it's a different kind of suppression or a different kind of exhaustion. And we think it's really mitochondrial metabolism or metabolism in general is at the heart of this dysfunction. And so around this time, we also seen this paper by Luigi Nezzi's group. And they found they were looking at CD8s as well, linking it with lipids in the tumor also. And they found in pancreatic cancer, in early pancreatic cancer, there starts to be this appearance of little lipid droplets. And then there was much more lipid droplets in late pancreatic cancer. And this was associated with an increase in Tregs in the tumor and a decrease in CD8 T cells. And when they looked at the CD8 T cells in the tumor of the pancreatic tumors compared to in the spleen, they found that they had much more lipid uptake. And when they treated them a similar way that we did with lipids ex vivo, that it decreased interferon gamma and granzyme B, and they also had metabolic defects. So this kind of, when we put all this together, it looks like cytotoxic cells, so both NK cells and CD8 T cells, can't really use lipids in high lipid environments like obesity and the tumor microenvironment. And we were not that surprised about for NK cells. It looks like NK cells are not as metabolically flexible as other cells, but CD8 T cells we know can use lipids. There's a lot of literature showing the importance of lipids and fatty acid oxidation for memory T cells. But we think in environments where lipids are in excess, that it's damaging to NK cells and CD8 T cells. And then at the same time, on the tumor side, it seems to be the opposite. So my friend Simir Beyaz done this paper when he was a PhD student in Nature, and he found that in the LGR5 model of intestinal stem cells, when he fed mice a high-fat diet, their intestinals, the LGR5 positive cells, proliferated more and became more tumorigenic, more stem-like. And he found that this was because they turned on lipid metabolism pathways, particularly PPAR. And this allowed them to proliferate more and metastasize. And at the same time, in Barcelona, this paper also came out showing that metastatic tumor cells, the ones that move from the tumor cells that escape the primary tumor and go to the secondary place, a marker of them was CD36, and which is a target gene of PPAR, and they use lipid metabolism possibly to escape or metastasize and survive in the other place. And so it seems that on one side, the cells that want to kill the tumor don't like lipids, whereas the tumor does. However, it's not all immune cells that can't use lipids. So Ping-Chi Ho last year showed this, and he found that as other people have also found, Tregs like lipids. And so in human breast cancer, he sequenced the Tregs in the tumor and in the PBMCs of the patients with breast cancer, and he found that metabolic or fatty acid metabolism was enriched in the tumor Tregs. They've also upregulated CD36 as well compared to in the blood. And then in the mouse model of B16, he found that tumoral Tregs take up much more lipids than Tregs in the thymus or the spleen or the lymph node. And when he specifically knocked out CD36 on Tregs, it prevented their accumulation or their excessive accumulation in the tumor, and it reduced tumor growth. And so we've also published this a few months ago, that another cell that seems to like lipids is those that produce IL-17. And so we studied this in gamma delta T cells. This is work done by Mathilde and Claire in my lab mainly, and although it was a big lab effort. And so in the gamma delta field, there's two types in mice, mainly two types. Those that express CD27, which produce interferon gamma, and CD27 negative, which produce IL-17. And generally, the interferon gamma producers are anti-tumor. They're quite like NK cells, and the ones that produce IL-17 are pro-tumor because they can attract neutrophils, which can help with metastasis, and they can be pro-angiogenic. And so when we looked at the two different gamma delta T cells in terms of metabolism, the main thing we noticed was the ones that produce IL-17 had loads of lipid droplets inside of them, whereas the other ones didn't, which is typical like NK cells and CD8 T cells. And so we found that when we looked at Bodipi palmitate, which is just, we just give this into the culture and see what cells take it up, the gamma deltas that produce IL-17 take up much more palmitate than the ones that produce interferon gamma. And when we just look at them resting, they have much more mitochondrial mass and mitochondrial potential. And so if we put them all together and activate them all together, you see some produce interferon gamma, and they are not the ones that are taking up lipids, while the ones that produce IL-17 are all taking up lipids. There's no real IL-17 produced in cells that are not taking up lipids. And so we thought that, you know, because they like lipids, they may be expanded in obesity where there's an excess lipids. And so this is just shown, you know, in general, they have much more lipid droplets compared to the ones producing interferon gamma. And Claire has done, she's still working on this, but so far we found that they take up fatty acids and cholesterol, and the lipid droplets are a combination of both triglycerides and cholesterol. And so we wondered in human and in murine obesity, where there's an increase in both fatty acids and cholesterol, what would happen? And so as we predicted, we get a big expansion of gamma delta 17s, and there's no change in gamma delta interferon gammas. We also found the same in human obesity, and other people have found it, Barbara Nikolajczyk and Andy Hogan, in many different cell types, mate cells, gamma deltas, TH17s, NKT17s, are all obesity in humans pushes cells towards being an IL-17 phenotype. And we also see, now this is outside the setting of obesity first, we also see that these gamma delta 17s are expanded in the tumor environment. So this, you know, usually the gamma delta interferon gammas are much more abundant in the body. And so, you know, it's around 30%, say, in the lymph node that are the IL-17 producers. In the tumor lymph node, it's not much different, but in the tumor itself, there's an increase. There's more than 50% of the gamma deltas are the interferon gamma, are the IL-17 producers. And so then we wanted to see, well, what happens if you combine obesity and the tumor? So we fed mice a high-fat diet or a standard fat diet and then give them B16. We found once again that the tumors were bigger in mice that were fed a high-fat diet, and there was a further expansion of gamma delta 17. So where it was around 50%, now it was over 80% of the gamma deltas were IL-17 producers, and there was a significant drop in the interferon gamma producers, similar to NK cells and CD8 T cells in obesity. And so we wondered if we were able to manipulate lipid metabolism, would we be able to reduce the amount of gamma delta 17s? And so one of the ways that we tried it was with orlistat, which is a lipase inhibitor, which blocks uptake of lipids or absorption of lipids. And so what we found was when we gave orlistat to the mice, they had a decreased accumulation of gamma delta 17s in their tumor, and the IL-17 producing gamma deltas had less lipids inside of them as well. And as a result, there was a decrease in tumor growth. But as we know, the tumors, this was just injection into the whole mouse, and tumors like lipids too. So this could have been, and most likely was, due to the tumor being inhibited in lipid uptake as well. And so we tried another way to see the effect of lipids on gamma delta 17s and tumor growth. So we expanded gamma delta 17s for adoptive transfer into the tumor, and then we did the expansion in the presence or absence of cholesterol. And so here is the tumor growth. And if we inject in regular gamma delta 17s, in red here, we see that they have slight increase in tumor growth because they can be pro-tumor, but ones that had been cultured in the presence of cholesterol, which produced more IL-17, had made the tumor growth accelerated. And so just to summarize that part, in general, there's two types, a pro-tumor and an anti-tumor gamma delta. The pro-tumor have a lot of lipid droplets, and they're expanded in obesity. They're also expanded in the tumor microenvironment. They're further expanded in the obese tumor microenvironment, and they can be inhibited by orlistat, partially inhibited, and they can be expanded by cholesterol. On the other side, you have the anti-tumor interferon gamma producing cells, which don't seem to use lipid metabolism. They're reduced in obesity, they're reduced in the tumor, and they're further reduced in the obese tumor. And so if we put all of this information together, what we think is that lipids are a common feature of the pro-tumor response. There's lots of other labs working on this, lots of other data to support this. So in obesity, you've got an increase in free fatty acids. It's coming from the diet, from adipocytes bursting adipose tissue, releasing free fatty acids. It goes to the liver, it goes to the muscle, the pancreas, vasculature, and this is associated with different diseases and insulin resistance. But we also know now that in obesity, the tumor can take up free fatty acids as well, and it can accelerate the tumor growth, proliferation, and metastasis. Where on the other hand, the immune cells can also take up this excess lipids, but it seems to impair NK cells, CD8 T cells, gamma delta interferon gamma positive cells, where it can fuel Tregs and gamma delta 17s, which can have a pro-tumor role. And so overall, lipids have a pro-tumor function through many different ways. And so one of the things that we're interested in is if we can target lipids or immunometabolic pathways, particularly lipids, to improve immunity in obesity, for example, against cancer and infection. And so the last part I'll talk about is all obesity or are all lipids the same? And so Hannah is a PhD student in the lab who has been working on this project. And it's been quite fun to do this project. There's some unexpected results. So here we wanted to see if we change the type of lipid that we give the mouse while still keeping the mouse obese, does it have any impact on the immune system? So here we fed mice animal fat diet, which could be from lard or butter, or a plant or nut high-fat diet, which was from palm oil or peanut oil or coconut oil. And so it was an isocaloric, they were all isocaloric diets. 45% of the calories came from fat in all the diets. And there was the same calories per gram of food, the same amount and type of carbs, proteins, vitamins, and minerals. So the only thing that was different was the source of fat. And so first thing we see is when you feed mice a high-fat diet, it doesn't matter where the fat comes from, they all become obese compared to standard fat diet. And when we looked at their fat pad weight, their visceral fat pad, they were all significantly more fat than the ones fed a standard fat diet. But when we did the tumor, then we did the B16 model, we actually seen that they had different effects on tumor growth. So this is the one that we have done many times, which is the one most labs use, the lard diet. And this accelerates tumor growth. The coconut diet didn't seem to that much. The peanut oil one did, it was a bit variable. But the two that were most different were butter and palm oil. So the mice fed the butter diet had much more accelerated tumor growth, which is even higher than in the lard diet. And this is even striking for B16, which isn't as much affected as MC38 by obesity. But the palm oil diet, now we've done this 4 times, the tumor growth is always just under the standard fat diet. And this is despite the mice being the same size. So here, if we just look at the tumor volume, we see much higher, much bigger tumors in mice fed butter versus palm. And so we're wondering, can we learn something from the worst versus the best? And what we're wanting to learn is not what fat we should eat, should we not eat butter? Should we eat palm oil? but more about, how could these different diets affect, what is it that's in them, or what effect is it having on the body, and how does that go on to change the tumor progression and the immune system. And so the first thing to look at is the fat distribution. So again, we fed the mice a high-fat diet. This is without tumors now. And again, the mice gained identical amount of weight, whether they were fed butter or PAM. And their gonadal adipose tissue and their subcutaneous adipose tissue was equally expanded in both butter and PAM. So it wasn't that it was like a kind of apple versus pear shape. They all put on the same amount of weight in the same places. And so doing the tumor model again, we see again the butter the butter causes accelerated tumor growth. And when we look inside the tumor at the TILs, we see that there's a decrease in NK cell numbers, a decrease in T cell numbers, but not in mice fed PAM. Now it is slightly reduced, the NK cell numbers is slightly reduced, but it's not significant. And then there was also a loss of function. So again, there's a total loss of function in the tumor anyway, but it was further lost in butter. So there was less interferon gamma, but not in mice fed PAM. And this was the same in CD8 T cells. There was reduced interferon gamma, but not in mice fed PAM, despite them being the same size, despite them being as obese. And so we wondered if there was a difference in the lipid uptake. And so we found that mice fed a butter diet had much more lipid uptake in NK cells than mice fed the standard fat diet or the PAM fed diet. And we found that this was the case in NK cells, but also in CD8 T cells and gamma delta 17s. This was the most striking result of all. When we did seahorse, we isolated NK cells from mice who don't have tumors that were fed butter or palm oil, and we found that, as before with lard, there's complete metabolic paralysis in mice fed the butter diet. There's no glycolysis or oxphos when they've been activated, but the mice fed the palm oil had the exact same metabolic response as the mice fed the standard fat diet, despite these being equally as obese as those fed butter. So this is now separating metabolism from obesity. And so we wondered if there's any major metabolic changes between these diets that we wouldn't have predicted. So what usually happens in obesity? So we can study this using the metabolic core and the metabolic cages. So we put mice into these cages, and it measures everything related to their metabolism and how much they eat and move and drink and their respiratory exchange ratio. And so what we know is this is the respiratory exchange ratio of oxygen to carbon dioxide. And so the measurement goes from 1 on the scale, which mice are burning carbs, to 0.7, where mice are burning lipids. And in between, it's a combination of lipids and carb burning. And so mice have a metabolic circadian rhythm. So at nighttime, when they're eating and they're moving around, they're burning carbs. And in the daytime, when they're asleep and they're not eating, they're burning fats. They're burning their fat stores to keep warm and for energy, and then so on. And we know from many different labs that the metabolic rhythm is lost in obesity. So there's a switch to burning fat all the time. So there's a decrease in the RER, and it doesn't change from day to night. And so we wondered what happens in whole body metabolism when mice are fed mice are fed butter versus PAM, sorry. And so here's the RER, and we find again that there was no difference between the two. So there's a loss of the metabolic rhythm, and the mice are burning fat in day and night. And there was no difference in the whether the diets were butter or PAM. We also wondered if this was a kind of a metabolically healthy obese. And so we predicted that the PAM-fed mice would not have impaired metabolism, but in fact they did. So the fasting glucose was elevated in both mice fed butter and PAM compared to standard fat diet, and they both had impaired glucose tolerance. And so there wasn't, we were quite surprised there wasn't any gross metabolic differences. They still lost their circadian rhythm of metabolism, and they still had an inability to handle glucose correctly. And so we wondered, was there more subtle changes in the systemic metabolome? And this is work that we did with Ivana Mills and Ed Chucheni. And so they looked at we looked at the we collected serum, and they did metabolomics, and we found that there was, there's certain metabolites that are always changed in obesity, aminobenzenoic acid, Hipparate, for example. It's probably a reflection of the reduced microbiome diversity. And here it didn't matter where the fat came from, there was reduced aminobenzenoic acid and Hipparate, despite the mice having despite the differences that we've seen in the immune system. But we did see some systemic differences in metabolites. We see that, so here it's shown as decreased in butter and increased in butter, because there wasn't really that many differences in the PAM versus the standard fat diet. So really decreased in butter is compared to PAM. So we did see that there was a decrease in TCA cycle and glycolytic intermediates in butter, which we've also seen in lard before, because there's a reduction in glycolysis and amino acid metabolism in obesity. And then we've seen an increase in medium long chain acyl carnitines in butter compared to standard fat diet and PAM. And so this is something that Hannah was thinking of. So, this suggests here the increase in the acyl carnitines suggests that what we could be seeing is incomplete fatty acid oxidation. And so just to recap what this might mean. So fatty acids, to be metabolized, they go to fatty acyl-CoA, but with the acyl-CoA can't go into the mitochondrial membrane. And so CPT1 takes the CoA off and swaps it for carnitine, and then we have the acyl-carnitine with the fatty acids. And then further, the acyl-carnitine has to be go into the inner mitochondrial membrane for oxidation, and this is done through CACT. And so in obesity, you often see an increase in CPT1 because there's an increase in fatty acids going into the mitochondria, but you also often see no increase in CACT. And so as a result, you get this accumulation of acyl-carnitines in the here in the cell, and then they're shuttled out into the serum. And this is a marker of incomplete beta oxidation. And so this paper is the inspiration for why we ended up thinking about this, which has shown very nicely that mitochondrial overload and incomplete fatty acid oxidation is associated with obesity. And so also the acyl-carnitine, the presence of acyl-carnitines in the serum can be used in the clinic as a marker of inborn errors of metabolism. And so we really feel that in butter and in lard-fed mice, there's so much going in here, and then they're being shuttled out because it's incomplete fatty acid oxidation. And this seems to not be the case in PAM-fed mice, but only in butter-fed and lard-fed. And so we wondered then, if you got this accumulation of acyl-carnitines, here octadecanol carnitine and myristal carnitine, what effect does this have on the immune system? So the first thing Hannah did was treated the T cells and NK cells with the different carnitines. So C8, a short one, a C16, which is the palmitate one, and C18, which is the one from stearic acid. This is the longest one. And she found that it had no effect on the viability, so it wasn't killing them. But the longer the chain, the more impact it had on the function. So C8 didn't have any impact on the ability of cells to produce interferon gamma, but C16 did in a dose-dependent way, and this was even worse with the longer chain C18. And so when we do by seahorse as well, we see the same. So sorry, here is just this is just expanded without stimulation. Black here is stimulation of the T cells with anti-CD3 and CD28. They've upregulated glycolysis and OXPHOS. When we give the C8, the acyl carnitine version C8, it actually boosted their metabolism in a way. But when we give C16 in red or C18, even further impairment in both glycolysis and OXPHOS. And so, what does this all mean? Well, first of all, this we think, because the longer the chain, the more the longer it takes to do to complete fatty acid oxidation, And we know in the butter-fed mice that there's more stearic acid, and so there's more longer chain fatty acids, and so this is leading to the accumulation, particularly of long chain acyl carnitines, which have a particularly detrimental effect on T cells. So what does it mean in the bigger picture? We don't want to be given a message that butter causes cancer and Pam doesn't, especially would never say that about Kerrygold Irish butter. But what we do like about these results is that they're a model for, you know, basically what we've found now is there's the same amount of fat in the diet. There's the same amount of adiposity in the mouse. The mouse gets exactly the same amount of obesity. But despite these things, you can have profound differences in the cellular and systemic metabolism. So also we see, you know, grossly, there's no big difference between these two. But if we look deeper, we see there's a big difference on the effect of these two diets on immune cell metabolism and on the metabolome. And so this is still a work in progress, but I think it nicely can illustrate that how systemic metabolism and the changes here may be related to the changes that we see in the immune system, and then the outcome for the tumor growth. So thank you to everyone involved, especially everyone in my lab, and to our collaborators, David Finley, Don Lochet, Andrew Hogan, Ivana and Ed, and Linda Sinclair and Doreen Cantrell in Dundee, who's done proteomics. Thank you all for listening, and I look forward to answering questions